today I have the honor to introduce our Symposium plenary speaker and our wildlife biology seminar speaker, Dr. Kevin Crooks. And I can say it wasn't very easy to get him here. If anyone has tried to get an email back from Scott Mills, you might have an idea of what I've been up against, but we're very glad that he was able to make it, and it was well worth the effort. So I want to tell you a little bit about his background. For some of you that were here last night, it might be repetitive, but I want everyone to know a little bit about him and why we brought him in here for this meeting. So Dr. Crooks did his PhD at the University of California at Santa Cruz with Michael Soule, and his thesis was on Mesopredator Release and biodiversity on fragmented landscapes. And today, Dr. Crooks is an associate professor and a distinguished Montfort professor at Colorado State University. What inspired us to bring him to this meeting is his obvious connection with connectivity conservation. So his work is primarily focused on anthropogenic impacts to wildlife populations, especially with respect to habitat fragmentation, urbanization, and connectivity. And even more inspiring to me is his obvious commitment to science with conservation consequence. So the application of science to inform conservation. And I'm sure that this facet of his work is going to shine through in his presentation today. And with that, Dr. Kevin Crooks.
Thank you. Thank you. It's a pleasure being here. I like the weather up here. It's very nice. I think Fort Collins is sunny at this moment. But thank you, Julie. She showed remarkable persistence to track me down and get me up here. I should say that I'm so impressed by this meeting that you guys have held through the Montana chapter of the SCB. Really, it's really amazing. All of you and all the research that's being presented, it's fantastic. So before I dive into the details of my research on connectivity and wildlife conservation and urbanizing landscapes, I first wanted to pull back, and maybe this follows from the panel discussion last night, and consider the impacts of humans on our natural world. And we know, of course, by sheer numbers alone, our impacts are great. We've all seen this figure. Humans have, the population bomb has exploded. We've experienced rapid exponential population growth over the last couple hundred years. So by sheer numbers alone, our impacts are great. Over the course of this century, our numbers will continue to grow. It'll asymptote out, hopefully, maybe around 9, 10, 11, 12 billion people. So the question is, where do all these people live? How do we fit all these people in? Well, one way to answer that question, this is somewhat dark, but is to look at the lights we create at night from space. And I'm sure that many of you have seen these kinds of photos. This is from a NASA website. And it becomes very clear that people live in major population centers. In fact, almost all of the expected population growth this century is expected to occur in urban areas. And demographers tell us that for the first time in human history in 2007, that the majority of the human population, over 50% of the human population, now live in urban areas. So a major demographic milestone. In fact, there's more urban residents today on the planet than there were residents on the planet when I was born in 1968. Of course, in the United States, continental United States is highly urban as well. These are night lights colored blue. 80% of our surface area, 80% of the population is urban. And over the next 25 years, the surface area of the continental United States that's impacted by urbanization or transformed to urbanization will increase by 80%. So what are the impacts? Well, we can see the man swarm, as Dave Foreman has recently called this. This is a population density model developed by a colleague and collaborator, Dave Theobald, at Colorado State University. It's basically population density per square mile over the last 200 years. And you can see the spread of humans across the nation and then the origination of our major metropolitan areas. You can see that here in the Rocky Mountain West, we were spared until the last 50 years or so. Well, the impacts are going to be great. We know that habitat loss and habitat destruction has been identified as one of the greatest threats to biological diversity worldwide. This is a now classic study by David Wilkov where they went into the Endangered Species Act and simply tallied up the threats to endangered species and found that habitat loss was the primary threat. Similar studies have been done globally with IUCN Red List data. And then Brian Czech did a follow-up study going back into the ESA, and this time subcategorizing different components or agents of fragmentation, including urbanization, and found that urbanization was a leading agent of fragmentation and a primary cause of species endangerment for U.S. threatened and endangered species. In fact, it was just second to non-native species. If you excluded Hawaii, urbanization for the continental U.S. was actually the leading cause of endangerment. Well, where do we expect the impacts to be greatest? Well, the answer is kind of obvious. Where there's the most urbanization and the most biodiversity. So one of those places is right here in coastal Southern California, which is a biodiversity hotspot and also one of the largest metropolitan areas in the United States. It has over 20 million people now. In fact, Southern California, and I'm going to be focusing on that today, is one of the world's epicenters of biodiversity as well as extinction and endangerment. Now, which species do we think might be most sensitive to habitat fragmentation, habitat loss, and urban sprawl? Well, it could be our largest bodied organisms, such as large carnivores, because of their wide resource requirements, low population densities, and also direct persecution by humans. So what I'd like to do today is talk a little bit about my work on urban carnivores. And what I'm going to first do is just give a real quick overview of the Southern California study system and some of the different survey techniques that we've used over the last, I'm going on almost 2 decades working down there, and some of the student projects that I've had over the course of the years. And then I'm going to tackle one specific question, which is to ask how carnivore species differ in their sensitivities to fragmentation. And a take-home message from this is that these large carnivores require connected landscapes. They require connectivity for persistence. So I'm going to end the talk by talking about the concept of connectivity conservation, which I thought was appropriate for the topic of the meetings, and then discuss 2 recent advances that we're tackling in my lab to assess and predict connectivity, and those are the development of movement models and habitat models. All right, so starting with the study system, I thought Michael Soule was going to be here, so some of these slides I threw in there for his benefit. But this is me in my younger years, when I first started graduate school with Michael back in 1994, and I'm standing on a suspension bridge over one of the San Diego canyons. These are patches of chaparral and coastal sage scrub habitat that have been isolated over different times over the last century of urban development. This is it from a much higher vantage point. And you can see this is San Diego and neighboring cities. And you can see the geography is basically, you have these broad coastal mesas that are dissected by these steep-sided ravines or canyons. And these canyons basically function, therefore, as habitat islands immersed within this urban sea. And those habitat islands differ in size, and they differ in the degree of isolation. So I've been interested in a look at how carnivore species and other wildlife can or cannot persist in this kind of system. So we use a variety of different survey techniques. I know that many of you also employ these same suite of survey techniques. I've tried a lot. We've used non-invasive tracks surveys, remote camera pictures. You're going to see a lot of those today. Scat surveys. I've had students do non-invasive genetic sampling from scats and from hair snares. We've done quite a bit of telemetry, first traditional VHF telemetry, now more GPS telemetry on both wild native carnivores and domestic cats. And then this definitely follows from last night's discussion. The primary driver in these urban systems, not surprisingly, is humans. And so it's become progressively clear to me that we need to understand the human dimension in order to promote and understand the conservation of these wildlife communities in these urban areas. So over the course of the years, I've become more interested in tackling this human dimension, particularly collaborating with people who actually know this, social scientists. So developing collaborations to understand better attitudes, knowledge, and education of these urban residents about urban wildlife. Okay, just threw in some camera pictures that you might need a little breather. And also it shows you that these remote camera surveys coordination with other kinds of survey techniques, maybe more invasive techniques, such as GPS telemetry, can help us answer some questions on some difficult to study organisms. So we can answer questions on large carnivores, such as mountain lions. I try to pick the cuter pictures for you. Bobcats. This is we have a lot of cameras stationed at underpasses under roadways, and so when you see these corrugated walls, usually those are wildlife underpasses. Here's 2 coyotes approaching one of the wildlife underpasses. We also get the mesopredators, uncommon mesopredators, relatively uncommon, such as badger and my favorite species on the planet, which is spotted skunks. But we also get the common mesopredators like gray foxes and striped skunks and raccoons. Now, sometimes things don't end up well for these mesopredators. This is a picture that we just got in August, and this is camera work that we're doing outside of Boulder, and we got a picture of this mountain lion heading right down to the urban edge of Boulder, and then like an hour later, heading right back with this raccoon. Intra-guild predation in action. We get pictures of course, the non-native exotic predators, such as this domestic cat going through an underpass with a prey species in its mouth on April 17th of 2009, and 2 days later, coming through with another prey species. Now, sometimes things don't turn out well for these exotic mesopredators either. This is a picture we got a couple weeks ago outside of Boulder, and I know it's hard to see. I thought I'd include it, though. This is a coyote, and that is a cat. And that coyote is about ready to pounce. Very rarely documented. But sometimes we see the end product of that interaction, such as in this photo. And this is, if you can't see it, a coyote carrying a cat through an underpass. What I like about this picture, there's several things, some I won't touch on, but what I like about it is that it encapsulates my research. We have trophic interactions and connectivity in a highly urban system. And then, of course, we get pictures of other large mammals, such as ungulates, but sometimes things don't turn out well for the ungulates either. This is a picture we got of a coyote carrying a deer head. And then sometimes the pictures are just downright disturbing. Nightmarish. Keep you up at night. But I ask you, what is more disturbing, this picture or these? And I'm going this. So it turns out that the same sites, same trails where we're putting these cameras, these wildlife movement paths, are human movement paths, or recreational trails. So, not exaggerating, we're getting thousands of pictures of wildlife, and we're getting tens of thousands of pictures of humans. Often Southern Californians, I'm showing you the G-rated pictures here. We can use this to our advantage. I've had a couple students look at the impacts of recreationists on wildlife activity, such as Shalene George. For example, she found that bobcats became more nocturnal in sites where there was more human recreation during the day. We're also using cameras to do non-invasive population estimation. I know quite a few of you are doing this as well. So this is a grid of cameras in Orange County on the coast. I have a couple of graduate students now, Robert Alonzo and Jesse Lewis, who are identifying bobcats individually by their pelt pattern. And then we can use a mark recite framework with GPS collars, or a mark recapture framework with the pelt IDs to estimate densities. I had another graduate student, Emily Rule, who used non-invasive genetic surveys. She developed a hair snare where she put a welcome mat carpet baited with a scent lure and a visual attractant. And the idea here is that the animals rub on the hair snares. So here's a bobcat, and here's a couple of coyotes. Here's a gray fox having a little too much fun on the hair snare. Mountain lions, skunks, we don't want them near the hair snares. And you know the idea that they leave their hair, we can amplify DNA, use mitochondrial DNA for species identification, microsats for individual identification. Emily published a paper a couple years ago where she used fecal DNA in a mark recapture framework to estimate densities of bobcats. And she's currently using the same data to estimate gene structure and genetic diversity, gene flow. Okay, so in terms of a question that I want to pull out from all of these surveys for this talk, I've been interested in evaluating, as I've said, if, how, and why carnivore species differ. So if we go from an attack landscape down to a landscape that's highly fragmented, can we predict which species we're going to lose in sequence? And we can. Habitat fragmentation, as you know, is a multi-scaled issue, so we've been surveying suites of different carnivore species across range of fragmentation. So actually, this is stemming from some of my PhD work with Michael. These are logistic regression models where this is probability of occurrence and this is patch size. So these are very large patches, and these are those urban canyons I showed you. And you can see that as patch size declines, you first lose mountain lions, then lose bobcats, and then lose coyotes. Cats are fragmentation enhanced. They have higher probability of occurrence in the small fragments because those small fragments are surrounded by houses and the cats come from the houses. Perhaps of more relevance for this session, this is now fragment isolation. So these are highly isolated fragments separated by a lot of urban matrix, and these are connected fragments. And what I want to point out is that mountain lions absolutely required connectivity. I did not detect mountain lions in any sites that were truly isolated. They needed some sort of habitat linkage. And bobcats also were sensitive, and even coyotes declined in too small sites. So we can put these area and isolation curves together, and here we're predicting mountain lion probability of occurrence, and you have these plateaus of occurrence in large and connected sites. And they decline to these basins of local extinction in small and isolated sites. And what becomes interesting is you can then compare across species. So this is mountain lions, and that's bobcats. And you can see bobcats occur in small sites, but only as long as those small sites are connected. If the small sites become isolated, we didn't detect bobcats. And then finally, coyotes occur across a wider range of sites, but will decline and will disappear in the smaller urban habitat canyons. And it's there where I looked at this idea of Mesopredator Release. And again, I put this in for Michael, but I'll just briefly just mention it. I'm not going to go into the details, but a take-home message, what we found is that where coyotes did decline, that contributed to increased activity of mesopredators, such as domestic cats, which exert strong predation pressure on prey species, such as scrub-breeding birds. This topic has recently been called trophic downgrading, and this was featured in Jim Estes and Michael's paper in Science this year. Okay. So in terms of my trajectory for my research, this is the question I was tackling for my PhD, and it became patently obvious that connectivity was important. And so I became more and more interested in the concept of connectivity conservation. So what I want to do now is overview that and then talk about our movement and habitat models. So there's many different definitions of functional connectivity. This is probably one of the most well-cited definitions. This comes from the landscape ecology literature by Phil Taylor and colleagues, and it's the degree to which the landscape impedes or facilitates movement among resource patches. And the critical component of functional connectivity, as you all know, is movement. The more movement, the more connectivity. The less movement, the less connectivity. It could be movement of organisms or genes or diseases. Many different studies have shown, including the work I just showed you from my work in Southern California, that where connectivity is lost, species disappear. And therefore, the vision of connectivity conservation, of having connected landscapes, is a compelling one. But in reality, the practice, as we know from these posters and talks over the last couple of days, the practice of connectivity conservation is not simple. And this is part of what we discussed in the edited volume that Sanjan and I worked on that came out several years ago. So in terms of how we address issues of connectivity, I wanted to discuss two. And one is agent-based models. These are sometimes called individual-based models. And this is work that's been spearheaded. Really, the architect of these models is a former PhD student and postdoc in my lab, now a scientist in Southern California with the USGS, Jeff Tracy. And this is in collaboration with Jun Zhu, who's a statistician at the University of Wisconsin-Madison. And the idea of this is that these are agent-based models where the agent is the individual animal. The animal moves through the environment. It senses the environment. It makes decisions about what it senses, and those decisions can translate into behaviors. And then we can measure those behaviors in the field, and we can also model those behaviors. These individuals interact, and we can keep track of interactions with individuals and landscape elements, such as habitat patches, or between individuals, or interactions between individuals and pathogens. And for this reason, these agent-based models have been called computational laboratories, or sometimes called in silico experiments. The idea basically here is that you can manipulate components of the system to answer questions, for instance, about connectivity that would be impossible to do in the real world. So the focal species for these agent-based models are the large felids to assess connectivity at a regional scale for pumas and a local scale for bobcats. This is a large collaborative project working with many different researchers conducting GPS telemetry, on this case on bobcats in Southern California in Orange County. Also doing collaborative work with researchers doing GPS telemetry on pumas, such as Walter Boyce at the University of California, Davis, who has amassed a very large database on GPS telemetry movement of mountain lions, really throughout the South Coast ecoregion. So these GPS telemetry collars, sometimes they're programmed to take locations every 15 minutes or every hour, depending on the model, but they're really providing unprecedented data on the day in the life of one of these cats, how they manage to navigate through these highly urban landscapes. Now I have an animation, and let's see if it works. That mountain lion is moving. So this is an example of some of those move patterns. This is for a mountain lion in the Santa Ana Mountains, bordering new development in Orange County, and this is over the space of a couple days. And you can see the interesting movements this animal is making, right? It's coming right along these urban edges. So if we make a cartoon of what we just saw, in red is the actual animal's movement path. We don't know that. What's being approximated by the GPS collars are white, which is the approximated movement path, with each dot being a GPS location. And what Jeff Tracy is doing in collaboration with June Zhu is basically coming up with quantitative descriptions, statistical models to describe this approximated move path. So like the step length, which is the time or distance between successive locations, therefore the rate of movement. And then also turn angles, or the distance in direction to landscape elements like urban edges. Basically coming up with quantitative rules of movement, of how these organisms move. So to give you an example of how we might model movement response to a gradient of urbanization, with natural being in green and urban being in red, successive GPS telemetry points with response angles here. The dotted lines are the perpendicular gradient, uphill gradient of urbanization. I'm not going to get into the details of all of this, but basically Jeff models the movement angles with a von Mises distribution, which is basically a circular analog of a normal distribution. And the steps here is that he parameterizes the models with maximum likelihood approaches, uses AIC model selection to pick the best models, and then uses those best models to simulate virtual animals moving through the landscape, moving the way that the real animals do. So here's an example I'm going to give you for one animal, and this is animal B10. And what we see here in green is the distribution of move directions for this bobcat B10 as the percent urbanization around each GPS location increases. Okay? So the idea here is that if the animal is moving away from the urban gradient, we're going to see avoidance in the move angle. Okay? So moving in this direction is going to result in less urbanization at the next location. If the animal is moving towards the urban gradient, moving in that direction, of course, is going to result in more urbanization at the next location. And then if it goes parallel, left or right, it's moving parallel to the urban gradient. It results in about the same amount of urbanization at the next location. So what this animation is going to show you here is how the percent urbanization around each point, as it increases, how is the animal responding? And you can see as the percent urbanization increases, as the animal is getting nearer and nearer the urban edge, it starts to avoid the urban edge more and more, starts to turn away from it. Around 20%, you really see some kind of threshold where it really starts to avoid it. So this is from the empirical data. So the take home here is that if there's very low amounts of urbanization around the animal, there's little to no response to urbanization. if there's a moderate amount of urbanization, the maximum percent urban we recorded for this bobcat was 44.8%, basically right on the urban edge. The animals strongly avoid it. All right, how do we apply these kinds of move models? Well, one thing we can do is go to that animal's home range and simulate these virtual animals moving through the home range. So what I'm going to show you here is 50 virtual bobcats who move very quickly. Wow, moving quick. So 50 virtual bobcats following the best approximating model about how bobcat B10 responded to urbanization. 50 simulated movements every 15 minutes, which is the move step for the GPS collars that we collected for B10. And what you get basically is a prediction of movement, which is a layer of movement, which is really a predictive surface of functional connectivity. Now, the potential power of these agent-based models is that we can simulate movement not just where we collected the data, but we can also potentially simulate movement throughout a whole study area to assess connectivity. So one way to do this that we're trying is to identify different core areas. These are large natural areas with little urbanization. And what I'm going to show you is some simulations. We have 1,000 of these virtual bobcats with 5,000 simulated moves each. For the first iteration, the baseline iteration, they're not going to be able, if they hit the Pacific Ocean, they can't surf. They can't swim. They're going to stay on land. So, we have all the virtual bobcats moving around. There's 8 different core areas. This one right here is the Santa Ana Mountains. This is basically the primary core area in the region, and then these are all satellite cores. And as you can see, as the simulation progresses, you see more intermixing of individuals, which are individuals moving between core areas, which is functional connectivity. So this is the end of the simulation, and then we can actually quantify this by simply just counting the number of individuals that moved between each pair of core areas. And you can see that for the Santa Ana Mountains, there's pretty good predictions of functional connectivity between 2 and 4 and 5 and 7. Okay? But what did I not include in that layer? I didn't include roads. And roads are a major barrier to bobcat movement. They're the primary source of mortality of bobcats in this area. So what Jeff did on the second iteration is now added the major roadways in the area and then run the model. And you can immediately see that there's a lot less mixing between the core areas, less functional connectivity. So this is quantifying the degree of movement of individuals, and red here is where there was movement before roads, and now there's no movement. So roads effectively severing the Santa Ana Mountains. But what can we do in the real world about this? Well, I was just talking about this over lunch. We can put in crossing structures. And what we are doing in the real world is working with the California Department of Transportation and the Nature Conservancy and other local agencies to site places for underpasses, install underpasses, and then monitor the underpasses. And these underpasses do work if they're put in the right place. What Jeff did in this virtual world is he basically perforated the roadway with virtual underpasses between 8 and 4. And so you could test to see whether or not these underpasses facilitate functional connectivity. And if we let it run long enough, what you will see is that you do have some bobcats finding these underpasses and using them. And that's showed here in the matrix where between 8 and 4, where there was no connectivity, now there is connectivity with the roads, with the underpasses. And so these are games that we can play, but we think they're not just games. We think that they can help guide real-world management and conservation decisions. One other analysis that Jeff did was to use his move models to be a proactive tool. Can we actually predict landscape connectivity in the future? And to do this, what he did was he simulated movement of now pumas across the entire ecoregion of Southern California. Gray is urban and white is basically open space, natural areas. And then simulated on a worst case landscape, which I call the nuclear option. And basically it's all private land being developed, and the only thing that's left is public land. And the future of Southern California is headed this direction. Right? So what you can see here is the degree of movement of the simulated animals between the major core areas for pumas in Southern California. A fair bit of connectivity between core areas now, but in the worst case scenario, you see where connectivity is lost. And what we envision here is that we can, again, use this as a proactive tool to focus our conservation efforts now in places that we predict might be of conservation concern in the future. Jeff's also been using these models for disease transmission studies. This I'm only going to go through briefly. This is funded work through the NSF Ecology of Infectious Disease program, where he's evaluating the effects of urbanization on disease dynamics in wild and domestic felids. And the modeling question here is, how does movement behavior interact with habitat fragmentation to affect contact rates between individuals and therefore disease spread? So this is what Jeff did for his postdoc work. And again, these are these computational laboratories, these agent-based models. So we varied parameters such as disease, the proportion of initially infected individuals, and the probability of disease transmission. Varied parameters of the landscape, the amount and degree of fragmentation of habitat. And varied properties of movement, the degree to which the matrix was permeable to movement, and different kinds of move rules. For instance, whether or not the animal exhibited point attraction, like a home range, or directionality, like a dispersing individual. In these initial simulations, and this work's in review now, he explored a very wide parameter space, 4,200 parameter combinations, and overall the simulations, 8.8 trillion moves, so it was very computationally intensive. To give an example of how we can simulate degree of fragmentation, simulated movement on different landscapes that differed in fragmentation, from landscapes with a lot of habitat remaining with relatively low fragmentation, down the process of fragmentation to areas with very low amounts of habitat remaining, and that remnant habitat is very parcelized. In these kinds of agent-based models, we need to keep track of not just the agent's location, like we did with the connectivity models, but also the agent's state, whether or not they were infected or not. Infected here is purple. At each move step, all the animal's neighbors are identified in a contact network. And then through the course of the simulation, we can keep track of the spread of disease both within and between patches. So each color here is an individual who was initially infected, and each node in this graph, this network, is where it transmitted a disease to another individual when it ran into it. And so we can therefore keep track of within and between patch transmission as fragmentation changes. I'm not going to go into the results here, but the take-home message, I think, from this was that movement was critical. It's often not incorporated in traditional disease models, but it was a critical predictor of disease transmission. And that's a bigger point I wanted to make, is that returning to the Taylor et al. Definition of landscape connectivity, movement among resource patches, I also found this other quote in that same paper where he said ignoring movement can lead to incorrect and potentially devastating consequences for conservation. And we're definitely finding understanding movement is critical to understanding patterns of connectivity and disease. Okay. So I'd just like to end by talking about another approach that we're trying out, and these are a development of habitat models at regional and global scales. This first are regional habitat suitability models for pumas, and this is work done by another postdoc, Chris Burdett, in my lab, in collaboration with Dave Theobald at CSU and others. And what we're doing here is returning to this same telemetry data that I showed you. This is the puma telemetry data that we use for the agent-based models. Now we're using for habitat models. We had a set of a priori models which evaluated hypotheses about how both natural and human landscape features affected puma habitat use. Again, using AIC to pick the best models, and then use those best models now to predict habitat suitability. And so here are the habitat suitability models that Chris generated. This is work, I should say, that was published last year in Ecosphere, which is ESA's new online journal. And these are the suitability models here. These are the telemetry data. Blue is low suitability, and like yellow and red are high suitability. The power of this is that we then can intersect these habitat models with Dave Theobald's urban growth models that he's developed not just for California, but he's actually developed them for the entire U.S. And so this enabled us to predict how urban sprawl will impact puma habitat and connectivity and fragmentation. A take-home from this work is that from these models, they predict that by the year 2030, starting at 2000, by the year 2030, an additional 10% of the study area will be transformed to urbanization. Recognize that by the year 2000, this is already a highly urban area. And that 32% of the suitable habitat for pumas on private land will be lost because of urban sprawl. Okay. And finally, we're developing habitat models to not just look at the regional scale for a single species for pumas, but we're now trying to go global with it and developing habitats or applying habitat suitability models for all terrestrial carnivores globally. This is 246 species and 13 families, and the way we're doing this is working with some habitat models that have been recently developed by the IUCN. And this is work spearheaded by Carlo Rondinini and Luigi Boitani in Italy. And so there are collaborators on this project. And what these large teams did at the IUCN for all mammals, so over 5,000 mammals, is they developed habitat models. And so this is a habitat model for an example. It's the Patagonian weasel. And what they're trying to do is move away from simple geographic range models and try to understand a little bit more about distribution within those ranges. This is an expert knowledge base combining with GIS layers on land cover, elevation, and hydrology, and it basically results in these habitat suitability models where you have suitable and unsuitable habitat. To give you a little bit more perspective, I'm going to zoom in here on one species, Canada lynx, and its habitat suitability model. Zoom in even farther into the Edmond-Calgary area. And the idea is for each of these species, they've identified low, medium, and high suitable habitat, and also whether or not the area is protected, which is in purple, and not protected, which is in green. Basically, the darker colors, darker green, darker purple, are high suitability habitat. So what Chris Burdett and Dave Theobald and I then did was calculate fragmentation and connectivity indices for the high-quality habitat patches for all of these species. Here's an example of the fragmentation index we use, which is called GIS frag. It's basically like a core area metric fragment. It measures the distance into high-quality habitat patches, so from the boundaries of the high-quality patches into the interior. So the idea here is that the higher distances, the darker colors, means that there's more interior habitat, less fragmentation. Okay? The shorter distances, meaning that there's less core habitat and more fragmentation. We then summed all of these indices up for all of the terrestrial carnivores globally, and what that enabled us to do is to generate these maps that are these global fragmentation hotspots. And this is published as a part of a session in the Philosophical Transactions of the Royal Society that came out in August. So the idea here is that in green are areas with a lot of core habitat. So this is with high carnivore species richness, and those carnivore species tend to have a lot of core habitat, low fragmentation. On the other end of the spectrum is black and red. These are sites where there's highly fragmented habitat. What we found from this work is that on average across all these carnivore species, only 54.3% of the range was comprised of high-quality habitat. Most of these macroecological hotspot studies that have been done focus on geographic range, and this points to that's insufficient because only a fraction of that's suitable. We also found, and of concern, is that 5.2% of the range, only 5.2% of the range was comprised of high-quality habitat in protected areas. Did I say I was optimistic last night? We also found that endangered species had higher levels of fragmentation. So this is IUCN status for the terrestrial carnivores. This is fragmentation index, high fragmentation, low fragmentation on a log scale. And species that were endangered and vulnerable had higher degrees of fragmentation of their high-quality habitat. Well, we did the same thing for connectivity. And basically what we did is not measured the distance within patches, but measured the cost-weighted distance between high-quality habitat patches. And the idea here is basically that the higher the cost-weighted distance, that means it's more isolated, and the lower the cost-weighted distance, that means it's less isolated. And the cost weights were based on whether or not the matrix was medium suitability habitat or low suitability habitat. Again, we can sum all of these up across all terrestrial carnivores, and now we're looking at global connectivity hotspots. So green here are areas with relatively high connectivity, but I'm going to particularly point out here the red and black. And those are areas that have high species richness, and most of those species have relatively low connectivity. Okay? So in terms of focusing our efforts, these would be sites that we would want to restore connectivity, and these are sites that we just want to conserve what's there. Okay. So ultimately what we're hoping is that with these field surveys as well as this connectivity modeling, that we can identify local and regional and global hotspots of fragmentation and connectivity. We anticipate that we can help, therefore, develop more effective conservation strategies for mammalian carnivores, species that we know are sensitive to fragmentation and that require connectivity. Okay. So with that, I'd just like to thank everybody. This is a huge team effort, and I'm a spokesman for a lot of work. Graduate students, postdocs in my lab, collaborators working on bobcats and pumas in the field, other collaborators on the other projects I told you, institution and support. Again, I'd like to thank and say how impressed I am by the Montana chapter of SCB, and also thank Michael, Sanjan, and Scott. And then I've also put my contact information here as well as those of my postdocs if you want to talk to them about more details. All right, thank you. Okay, so we have some time for questions. Any questions? Yes, in the back. Is that Scott? And I can repeat the question.
The low suitability habitats essentially became oceans in your model of connectivity, right?
That's right. The way that yeah, that's right. The way.
So you made a pretty big jump from suitability in terms of persistence of populations to suitability in terms of movement, for which you have some basis from all the field work over time. But how do you feel about making that transition?
Yeah, so the question was about whether the degree to which low suitability habitat is low suitability habitat and basically treating low suitability habitat as oceans. And the way that it's actually, it's kind of a misnomer about how they defined it. Low suitability habitat under their categories meant that the animal didn't occur there, that they weren't expected to occur there at all. Medium suitability habitat was that they could occur there, but they wouldn't reside there. And then high suitability habitat was that they could live there. And so we felt pretty comfortable saying, look, the low is just basically not habitat for them at all. Yeah. Yes. Who are you? Okay, yes. Sanjan.
So I really like those maps that you showed towards the end where you take your work and then apply it to a much bigger sort of continental scale. So look at lots of different carnivores. But one thing that struck me was that if you really look at just biology, if you look at what you know. So if you go to India, you will see wildlife, you will see lots of big animals, still fairly commonly. Like it's as easy to see a tiger in India as it is to see a grizzly bear in North America. Probably easier. Easier to see an elephant in India as it is to see wild bison in North America. So what I'm getting at is I wonder if connectivity and fragmentation is different in different continents, right? So a road in the US is very different from a road in India, where anything could cross it and probably not get hit. And fragmentation in Brazil just looks different than fragmentation in the US, because otherwise you can't look at that map and make sense of what you know exists there today. Because if you look at that, you think there's nothing going to be around carnivore-wise in Asia, in India certainly, or in East Africa, There are two areas I know pretty well, and yet I know from being there, you actually see a lot of wildlife. I mean, I'm not saying they're out of the woods or anything,
Right,
in
yeah.
great peril, but they're there, they're present, they move through landscapes. So this, so anyway, I just wondered if you could comment on that.
Yes. And so to repeat that is the degree to which these maps might mesh with what we see on the ground and actually represent threats to the animals. And I mean, it's a good point. Fragmentation does differ globally, but all in terms of developing the habitat suitability model, they're all using the same layers. So in terms of how these habitat suitability models were derived, they should be consistent no matter what the region was. So a road was a road was a road, or urbanization was classified the same globally. So I think that might help address that. But I do agree that translating this map to on-the-ground status is difficult. I am struck by the fact that our fragmentation indices did predict IUCN status. So there's some linkage there between what we're measuring and what's happening to animals on the ground. Maybe it's not now, but maybe it's in the future. I think a further step here, and we point this out at the end of the paper, is that what this doesn't do is it doesn't take advantage of the resolution of these models. So the scale of conservation action is on the local level. And so I think the next step here is actually dive down into regions where people are actually working on these animals and have empirical data on their distribution and movement, and see the degree to which they do a good job in predicting what's happening out there. We actually did that with Chris Burdett's Puma suitability model. So we overlaid the Puma map that we got from IUCN to what he derived for pumas in Southern California. It actually matched pretty well, except it over-predicted. It was liberal in what it was predicting where pumas were. Our actual empirically based habitat maps were much more restrictive in terms of where pumas were. Yes?
I was going to ask, it looked like just in some of the maps that some of the fragmentation is probably just natural fragmentation due to terrain or whatever, and then some of it is probably human caused, and if you get a sense of how that balance is playing.
Right. So the question is the degree to which the fragmentation that we're measuring here is anthropogenic versus natural fragmentation. And we didn't measure that, except for the habitat suitability models incorporated human land use in their suitability scores. So the matrix, the classification of the matrix between the high quality patches would represent the degree to which there was urbanization in between or human development in between. But that's a very good point, is that what we're measuring here is distributions of high quality habitat patches which could be separated by valleys or by Los Angeles. Yeah. Is that good? Yeah. Well, thank you very much again. All right, I'll be around this afternoon. Thanks.
Thank you very much, Kevin. I appreciate it.